press the bell icon on the YouTube app and never miss another update.
Namaste children. In our previous class we discussed about the central nervous system and also reflex actions. In this class we will discuss about the structure of eye and the mechanism of vision formation. Understood? So, many of the organisms have some special structures or organs that are able to detect them what all the changes that occurs in their surrounding environment. So, these structures are called sense organs. So, through these sense organs the organisms are able to detect the changes that occurs in their surrounding environment. So, the sense organs, how they receive stimuli, based on that we have three types of receptors, that is exteroreceptors and interoreceptors. Exteroreceptors and interoreceptors. Exteroreceptors means here the sense organs receive stimuli by the external environment. In the exteroreceptors, the sense organs receive stimuli from the external environment as they are in contact with the external environment. In this we have eyes, ears, nose, tongue and skin. All five important sense organs come under exteroreceptors. So, this classification previously, there are five types of sense organs in human body is given by Aristotle. He gave these five organs: eyes, ears, nose, tongue and skin. They are all belongs to exteroreceptors. Interoreceptors means here the sense organs receive stimuli by the internal environment, by the changes that occurs in the internal environment. Those are called interoreceptors. Example for this we can see pneumotoxic center are able to detect concentration of carbon dioxide in the blood. They all come under interoreceptors. And ear and eyes can also classified under teloreceptors because they are able to sense the stimuli from far away places also. So, this is about the classification. Then we will go for structure of eye. Today we will discuss structure of eye. So, the diagram will come for 5 marks and So paired eyes are present in skull in the region called orbits. They have three layers. Eyes are spherical in structure and they have three layers. Outer layer is sclera and the next layer is choroid and the third layer is retina. Outermost layer is sclera. First layer is sclera, second layer is choroid and third layer is retina. This choroid is pigmented area. What we see color in the eyes is due to pigmented region called choroid. And the retina is richly supplied with blood. And also choroid is also richly supplied with blood. And the retina, you see the photoreceptors. And the this is vitreous chamber, or you can aqueous So this is structure of eye. So it is spherical in shape and it contains three layers. Outermost sclera is dense connective tissue. The sclera, which is the outermost layer of the eye, it contains dense connective tissue and it is avascular. Sclera anteriorly forms a structure called cornea. Sclera anteriorly forms a structure called cornea and this cornea is avascular. There is no blood supplied to cornea. Hagagi eye donate maduvaga there is no rejection. Yake organ reject agalla andre the cornea is avascular. As it is avascular, as it does not contain blood, there is no antigen and antibody reaction. Hagagi there is no rejection when we do transplantation of cornea. Understood? Nimi competitive why there is no organ rejection takes place during the organ transplantation of cornea andaga because cornea is avascular. Understood? And the next layer forms a choroid. Choroid is a pigmented layer and it is thin in its posterior two-third and anteriorly it forms a structure called ciliary body. The ciliary body continues to form a structure called iris. It forms a structure called iris. This iris holds the lens in a proper position. This iris holds the lens in a proper position. And lens is a crystalline, transparent, present at the center of the eyeball, and the part of the iris which is covered the lens is called pupil. The part of the iris which covers the lens is called pupil. This part is called pupil. The diameter of the pupil will change when we see far objects or near objects. The focal length is adjusted by the contraction or relaxation of ciliary body. That we will discuss in the next. So, just the fluid which is present between the lens and retina is called, the chamber is called vitreous chamber, which contain a fluid called vitreous fluid, and the fluid which is present between the cornea and lens in an aqueous chamber is called aqueous fluid. So, the aqueous chamber contain a fluid called aqueous fluid and vitreous chamber contain a fluid called vitreous fluid. And this contain aqueous fluid. This aqueous fluid, it is watery in nature and the vitreous fluid is transparent and it is gel in nature. Transparent and gel fluid present in vitreous chamber is called vitreous humor antanu heltare. It is also called vitreous humor. And this is also called aqueous humor. And we see a blind spot and one more region, fovea. So, blind spot andre it is the area where optic nerve leaves the eyes. Optic nerve leaves the eyes and retinal blood vessels will enter the eyes. So, blind spot is an area where the optic nerve leaves the eyes and retinal blood vessels will enter the eye. So, in this area there is absence of rods and cones. As there is absence of rods and cones, there is no vision formation here. That is why it is called blind spot. Competitive will tell why that area is called blind spot, because there is absence of rods and cones, and it is the area where optic nerve leaves the eye and retinal blood vessels enter into the eye. And one more area is there, fovea. It is this area, fovea contain only cones. It contain only cones. In the retina, yellowish pigment area is present. In the retina, I don't have yellow chalk. So, in a retina, yellow pigmented area is present. That area is called Macula Lupia. That area is called Macula Lupia. So, in the Macula Lupia we see a depression called fovea which contain cones. So in this area, as it contains only cones, the resolution of image will be very high. Resolution will be very high in the fovea region because it contains only cones. So this is about structure of eye. So ನೀವು diagram 5 marks diagram ಇರುತ್ತೆ. So once I will explain. And so before going for photoreceptors, once I will explain. The eyeballs are spherical in shape, consist of 3 layers. Outermost layer is sclera, which is avascular, and inner pigmented layer, which is richly supplied with blood, is choroid, and also retina is also richly supplied with blood, that is innermost layer. The sclera anteriorly forms cornea, choroid layer anteriorly forms ciliary body and iris, and the retina is the innermost area where we see photoreceptors. It contains photoreceptors, and the chamber between the lens and retina is called vitreous chamber, which contain a transparent gel-like fluid called vitreous humor, and the chamber present between the cornea and lens is called aqueous chamber. It contains a watery fluid-like substance, that is aqueous humor. And the region which is the lens which is covered by iris, that part is called pupil. So the diameter of the pupil is controlled by, it is regulated by ciliary body. And this retina contains three types of cells from inner to outside. Inner to outside means inner it contains ganglion cells, middle it contains bipolar, and last it contains photoreceptors. Inner to outside, retina contains ganglion cells, then bipolar cells, and photoreceptors. See, inner to illinda. So first it contains ganglion cells, the next it contains bipolar cells, the next it contains photoreceptors. That includes 2 types of cells, rods and cones. That includes 2 types of cells, that is rods and cones. I will rub these labelings. There are 2 types of photoreceptors. One is rods, another one is cones. This rod is responsible for dim light vision. It is responsible for dim light vision. It is also called twilight vision or scotopic vision. And rods made up of a protein called rhodopsin, and it contains purplish red rods. Contain this rhodopsin is a purplish red protein. Rods contain a purplish red protein called rhodopsin. Rods contain a purplish red protein called rhodopsin, and this rhodopsin is having a derivative of vitamin A. It is having a derivative of vitamin A. Rhodopsin is also called as visual purple. It is also called as visual purple. The rods, they are responsible for vision during dim light. So that dim light vision is also called twilight or scotopic vision. And rods contain a purplish red protein called rhodopsin. It contains a derivative of vitamin A, and this rhodopsin is also called visual purple. Deficiency of vitamin A results in night blindness. Deficiency of vitamin A results in night blindness. Deficiency of vitamin A results in night blindness. Next, coming to cones. We have three types of cones. They have their specific photosensitive pigments. So we have red cones, so we have three types of cones: blue, green and red, and cones are responsible for daylight vision. They are responsible for daylight vision. It is also called photopic vision. It is also called photopic vision, and they have the different wavelengths. Blue is having nearly 480 nanometer, that means shorter wavelength. Green is having nearly 530 nanometer, wavelength of 530 nanometer, that is medium wavelength it is having. Red is nearly 580 nanometer, it is having a greater wavelength or higher wavelength. We get different colors based on the combination of all these three cones. So we get different colors, we are able to see different color vision mainly due to the combination of these three cones. And all these cones are stimulated, when they are stimulated with equal wavelength, when they are stimulated with equal wavelength, we see white color. We see white color. So cones are responsible for color vision. Why they are responsible for color vision? Because they contain three different types of photosensitive pigments, that is blue, green and red. They are, since blue are able to see in the shorter wavelength 480 nanometer, and green color is seen in the medium wavelength like the range 530 nanometer, and red is seen 580 nanometer. When all three cones are stimulated with an equal wavelength, we see a color called white color. That's the difference between rods and cones. These photoreceptors are present in retina. They are present in retina. Understood? So 2 marks difference keltara. Write the difference between rods and cones. Rods, they are responsible for dim light vision. It is also called twilight or Stereoscopic vision, they contain a pigment called rhodopsin, which is it is a purplish red protein, and it contains a derivative of vitamin A. In the absence of vitamin A, we see a condition called night blindness. Cones, they are responsible for color vision and also daylight vision or photopic vision. There are three different types of cones: blue, green and red. They have their own specific photopigments. When all three are stimulated with the equal wavelength, we get white color. We get different colors by the combination of these three colors. Understood? This is the difference between rods and cones. Next, we will go with mechanism of vision formation. Roughly, I will draw the diagram here. See, when the visible light, so when this visible light, the light with a wavelength of visible enters the lens, when it enters the lens through the cornea and aqueous humor, and then it enters and vitreous chamber, then it reaches the retina. Light rays, nodi, light rays, when light rays enters into the eye through the lens, they reach retina. Understood? Photoreceptors, they have when stimulated by light, then we see dissociation of opsin from retinal. There is dissociation of opsin and retinal. So hemoglobin ಯಾವ ರೀತಿ ಜೊತೆಗಿರುತ್ತೆ ಹಾಗೆ opsin ಮತ್ತೆ retinal ಜೊತೆಗೆ ಇರುತ್ತೆ. When it is stimulated by light, they are going to separate. Dissociation of opsin from retinal takes place. When the dissociation occurs, we see changes in the structure of opsin. That changes in the structure of opsin result in changes in the membrane permeability. That produces action potential, or they generates action potential or impulse. They generate action potential or impulse that is carried by bipolar neurons. Then from the bipolar, it will be carried to the ganglion cells. It will be carried to the ganglion. In the ganglion, we see collection of cell body and dendrites. Ganglion means collection of cell body and dendrites. Then it enters into the optic nerve. From the optic nerve, it enters into the visual cortex in the brain. Visual area or visual cortex in the brain. Where the impulse is analyzed and images which is formed in the retina is going to be recognized by the past experience and memory. Nodi, once again I will tell mechanism of vision formation. Mechanism of vision formation. How the vision is going to be formed? Usually we see the image which is formed on the retina will be inverted. Image which will be formed in the retina will be inverted. It will be inverted, but in the brain again it becomes upright. So whenever the rays enters into the lens to the cornea and aqueous humor, and it reaches the retina with a vitreous humor. Once it reaches the retina, it stimulates the photoreceptors present in the retina, that is rods and cones. When the photoreceptors are stimulated by light, that results in the dissociation of opsin from retinal. That results in the dissociation of opsin from retinal. Dissociate, separate ಆದಾಗ ಏನಾಗುತ್ತೆ? We see changes in the structure of opsin. Changes in the structure of opsin results in changes in membrane permeability. ನಿಮಗೆ ನೆನಪಿದೆನಾ? Generation and conduction of nerve impulse ಅಲ್ಲಿ at rest ಏನಿರುತ್ತೆ? Neuron at rest, inner it is having negative charge, outer it is having positive charge. So ಇದೇ ರೀತಿ ಇರುತ್ತೆ. When stimulus is not there, ಯಾವಾಗ stimulus ಬರುತ್ತೆ? When stimulus is applied, what happens? The stimulated area, in the stimulated area we see outer negative charges and inner positive charges. That results in generation of action potential, or that results in the generation of impulse that takes place when we see change in the structure of opsin occurs. So once the cells are stimulated by light, there is dissociation of opsin from retinal. That leads to changes in the structure of opsin. That changes in membrane permeability of photoreceptor cells, resulting in generation of action potential, or impulse is generated. That impulse which is generated in the photoreceptor cells are carried by bipolar neurons. They are carried by bipolar neurons. See, these are dendrites, these are axons of bipolar neuron. The dendrites receives the impulse and transfer that to the ganglions. All the axons of the cells of the ganglion join to form a optic nerve. They all join to form a, from both sides the axons of the ganglion will join and form a optic nerve. Through the optic nerve, the impulse will enter into the visual area in the brain, where the impulse is analyzed and image which is formed on the retina is going to be recognized by the past experience and memory. Understood? This is about mechanism of vision formation. ನೆನ್ಪಿಟ್ಕೋಬೇಕು ನೀವು. The image which is formed in the retina will be inverted. ಉಲ್ಟಾ ಇರುತ್ತೆ. It will be inverted. Suppose this is candle. So ಇಲ್ಲೇನಾಗಿರುತ್ತೆ? The image will be inverted in the retina, but brainally when it is analyzed, again it becomes upright. Again it becomes upright. So this is about the mechanism of vision formation. Understood? So idu nimage 3 marks barutte. Sometimes they may ask for 5 marks also. Understood? Next we will go with some points which you have to know, but they are not given in your NCERT. But ನಿಮಗೆ ಗೊತ್ತಿರಬೇಕು. Accommodation. So here the adjustment of, in the accommodation, the adjustment of diameter of pupil is done by the ciliary body. So this is lens, this is retina, this is lens is ciliary body. So when we see far objects, the lens will become concave and thin. This is for far objects. So lens will become thin and pupil diameter will be increased. Pupil diameter will be, see, the diameter of the pupil is increased by the relaxation of ciliary body. The muscles which are present in the ciliary body are going to be relaxed. That relaxation of ciliary muscles, will helps to increase the diameter of the pupil. So accommodation means the diameter of the pupil is adjusted by the ciliary body to see near and far objects. Is nothing but accommodation. What is accommodation? The diameter of pupil is adjusted by the muscles fibers present in ciliary body to see near and far objects. Is nothing but accommodation. Yenagatte, to see far objects, the diameter of pupil has to be increased. That is done by relaxation of ciliary muscles and the lens become concave and thin. So, to see near objects, this is ciliary body, this is lens, this is near objects. To see near objects, we need less diameter of pupil. So, then lens become biconvex and thick. Biconvex and thick, pupil diameter decreases due to contraction of ciliary body. This is nothing but accommodation. Understood? ನಿಮಗೆ ಇದೆರಡು ಏನು ತಿಳ್ಕೊಂಡಿರಿ ಅಷ್ಟೇ. Just what is accommodation ಅಂದ್ರೆ the adjustment of diameter of the pupil. What is pupil? The iris part which is in front of the lens is called pupil. The adjustment of diameter of the pupil to see near and far objects by the ciliary body is called accommodation. Ishtu gottidre saku. For the far objects, the lens become thin and convex. For the near objects, the lens become convex and thick. Ashtu nenpidre saku. The next we go with types of vision. Types of vision. We have monocular vision, binocular vision, and stereoscopic vision and telescopic vision. See, monocular vision antandre, in this type of monocular vision, eyes are present on the side of the head like in cow and horse. In monocular vision, the eyes are present on the side of the head, right side one eye and left side one eye. So, then they can see the larger objects on both sides. So, these animals can see larger area on both sides. That type of vision is called monocular vision, seen in cows, horse, and in many herbivorous animals we see monocular vision. Binocular vision, like in human beings, both the eyes are present in front of the head and both the eyes will see the same object. So, that is by means to both the eyes will see the same objects, that is binocular vision seen in human beings and most of the carnivorous animals, and in most of the carnivorous animals. Stereoscopic vision andre 3-dimensional view of the image is seen, that is called stereoscopic vision seen in human beings. Stereoscopic means 3-dimensional view of the images can be seen, that can be seen in human beings. And telescopic means we know in birds they are able to see far objects. So, that is about telescopic vision. So, this is about types of vision, and there are 2 types of defects. Defects of eye alli tumbane ide, but we will discuss only 2 types, that is hypermetropia and myopia. One is hypermetropia, far-sightedness, and myopia. Hypermetropia means they are able to see far objects. So, hypermetropia, the persons they are able to see far objects but cannot see, but not near objects, Istu nenpidre sa. In hypermetropia, the persons are able to see far objects but not near objects. Understood? In myopia, this is short sightedness. Here they are able to see near objects but not far objects, This is about myopia. Hypermetropia, it is far sightedness. So, far means they are able to see far objects but not near objects. Myopia, short sightedness, they are able to see near objects but not far objects. Understood? This is about the defects of eye. So, in this class what you have to remember means The structure of eye will come for 5 marks and mechanism of vision formation will come for 3 marks. And in your competitive exam, they will ask which layer of the eye is avascular. Which layer of the eye is avascular? Sclera is avascular. Which layer of the eye forms ciliary body? So choroid forms ciliary body. Which layer of the eye have photoreceptors? Retina is having photoreceptors. That is having photoreceptors. And fovea is present in which layer of the retina? Fovea is present in which layer of the eye? Fovea is present in retina. It contains only cones. Where the fovea is present? It is present in the yellow pigmented region of the retina. It is present in the yellow pigmented region of the retina that contain only cones. In this cones, resolution will be very high. And then blind spot is present in the eye where the optic nerve leaves the eye and blood vessels will enter into the eye. There we see absence of rods and cones. Hagagi, we will not see any image on that spot. That's why it's called blind spot. And there are 2 types of photoreceptors. One is rods, another one is cones. Rods are responsible for dim light vision, that is also called twilight vision or scotopic vision. Rods contain purplish red protein called rhodopsin, which is a derivative of vitamin A. And in cones, we have 3 types of cones which are responsible for color vision: red, blue and green. Different colors we get by the combination of these 3 cones, and when these 3 cones are stimulated with the equal wavelength, we get white color. Understood? And mechanism of visionally, when photoreceptors are stimulated by light, the dissociation of opsin and retinal takes place. That results in changes in the structure of opsin. That change in the structure results in changes in the membrane permeability. That membrane permeability results in the generation of action potential or in the generation of impulse. That impulse is carried by the bipolar neurons and it will be delivered to ganglionic cells. From the ganglion cells, it is carried through the optic nerve to the brain or visual area in the brain, where the impulses are analyzed and image which is formed on the retina is going to be recognized based on the past experience and memory. The image which is formed on the retina will be inverted, but the image which is going to form in the brain will be in upright position. Understood? That is about the structure of eye and mechanism of vision formation. So thank you. In the next class we will discuss about ear, structure of ear and mechanism of hearing. Understood?